Hello, everyone. Can you hear me? Is my setup working? It's been so long since I hooked everything up. So let me know if my audio is coming through. And I'll just drink tea for a little bit. Okay, I am heard. So that's good. Okay, there's a question already in the chat. Uh, what kind of spider is that on the title card? And that is Argiope, the yellow garden spider. They're great big spiders that show up in August usually. And they're all over the place in the fields here. They tend not to be found in town or near. What am I crunching on? Something was going crunch down under my feet. Anyway, they tend not to be in town or busy places like that because they tend to live on larger prey, things like grasshoppers and so forth. So if you've got a place with lots of grasshoppers, you might have lots of Argiope too. Okay, yes, you can understand the tea drinking. Yeah, I have switched away from coffee and I now drink tea almost exclusively. And it's necessarily hot because, man, it is cold outside. Ugh, really cold. Okay, so I'm supposed to start with my usual greeting. Hey, friends, good to see you all here. I'm back online after a very, very long gap in my schedule. And one of the things I got to talk about here is why? Why was I so slack? Yeah. Why was I not showing up at all? And why was my view count just declining? And why didn't I care and all this kind of stuff? Yeah, it's a little bit of a I'm just gonna make excuses for a little bit and then we'll start talking about the good stuff. Um, yeah, so I had kind of a crisis of confidence this past summer. Uh, One precipitating problem was, man, I had some bad knee problems. I spent most of this past summer in pain, just debilitating pain. It was not fun, and it kind of sapped my will and my interest in talking to human beings anymore. So I struggled with that. I'm getting better, started getting better near the end of the summer and into the fall. I'm still not 100%. Knees are creations of the devil, I have decided. They didn't evolve. They were put there by a satanic being. Because when they go, and they just do not let you do basic functions. So I was, I got to thank my wife who kept me going all summer long because there were basic things like there were days I could not get out of bed because that involved bending and straightening my legs, and she would help me out and haul me up and drag me off into my back room and throw me into a chair where I could just sit there and uh, feel sorry for myself all summer long. It was not pleasant. But like I say, I'm getting better now. Things are healing. I have, I still have problems, minor problems. I have named my knees. The right one is I've named achy, and the left one I've named wobbly. Can't trust either of them. And that's particularly bad now with the icy conditions we have now. I can walk a fair distance and I'm not in pain, but I just know if I slipped on the ice, I would be set back 3 months or so, and it would not be good. So I'm being inordinately cautious. Yeah. Okay, we got a comment. Fins are better than knees. I agree. They would be much better. Fins do fairly well. but you need a liquid medium for that. Anyway, so that's my self-pity session. I went through that. Then, there was just this growing awareness that here I was reluctant to get on to YouTube to talk and say things and stick my face out there. And I was realizing that, yeah, I'm getting older there. I was home to begin with. I'm getting worse. So I'm going to be a cranky, decrepit old man in no time at all. Well, I already am, but it's going to get worse. So that kind of sapped my will to do these public things. Um, Another thing is that I was on sabbatical, which was a good thing because I couldn't get anything done all of these last 6 months. Uh, and one thing I've noticed about myself is that if I stop public speaking, like lecturing in class, my voice starts to fade out on me and I have difficulty getting going. So that's another impediment to progress. So it's been difficult in that sense. And here's another personal problem is that my wife is going deaf and she can't hear me anyway. So it's a struggle that to say anything. I just feel like just pointing and saying, I'll go do this and you do that and keeping it to a minimum, which meant my voice was not getting any exercise at all. And even now, you can probably tell I'm a little bit raspy and hoarse because this throat has just been useless. I haven't been exercising it, which is another impediment to getting online and speaking about stuff. So that was a bad thing. Um, why am I getting back into it now? It's because I'm going to start teaching again in less than a month. So I better get back into practice speaking in particular about speaking about science. So that's one thing. I'm just got to get back into the grind and do this and get things moving again and get up and start, I don't know, voice exercises, singing la 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 or whatever, just to get the juices flowing. Yeah, you don't need to talk to spiders. Spiders are really nice that way. So, but I've got to start speaking to them more, I think. And drinking tea, which is good for your throat. Okay, so that's my side of things. That's why I've been a bad YouTuber. Uh, Another little problem, I have to be careful here because I don't want to sound like I'm criticizing other people for this, even though I am sort of. Um, I have become disenchanted with much of the YouTube science content. That a lot of it is okay. I want to say it's okay to aim your discussions at a general audience. That's really important. I think that's a good thing. We should do more of that. But too often I find what the science communicators online are doing is they're aiming it below the general audience. They are aiming their discussions at creationists. And I'm sorry, creationists are idiots. Really, they're uneducated, ignorant, know-nothings. And they're only arguing from a prior presupposition, and they're very annoying. So listening to some of the science stuff on here, there's occasionally really good stuff that I enjoy that's informative and useful. And then there's a whole bunch of stuff where I just want to scream at the screen that, no, why are you wasting your time with Cardova or Raw Matt or any of these people? Because they're stupid. They're not going to get your point. They are not motivated to listen and learn anything. So you're going to waste your time with these. And then to make it even worse, I look at some of these videos and they're 3 or 4 hours long. The science communicator in that discussion may be absolutely brilliant. I may enjoy listening to them and I want to hear more of them, but half the time is going to be spent giving a platform to a horrible, horrible, misleading disinformation specialist. And so you're basically promoting a creationist point of view. And I get so exasperated with that. Yeah, I don't want to hear that anymore. I want them to go. I mentioned I'm getting older, and really my patience is wearing thin. I just don't have time or interest in listening to creationist babbling anymore. I want them to go away. I want them to be disrespected and ignored. That's what needs to be done at this point, I think. So that was kind of bad. And hey, if you think I'm complaining about the science communicators, the atheism sites are worse. Seriously, why are we wasting our time with these kinds of arguments anymore? That the other side, the theists, they've got the same tired old arguments, not evidence, just arguments that they've been bringing up for centuries and that have been rejected and rebuted, rebutted and refuted over and over and over again. There's no further point. So, yeah, it just drives me up a wall anymore, which makes me just generally unhappy with YouTube. And let's not even get into YouTube algorithms and things like that that just promote the worst. Ah, okay, John St. Francis makes a comment. It's kind of a creationist one. Studying the complexity of cell biology changed my mind. Yeah, I think that the right approach isn't to say to dignify creationism with a response, but to go on and just say, oh, here's all this wonderful stuff that's going on in biology. Uh, that's what motivated me from the very beginning is I just wanted to talk about good science and all this exciting stuff that's coming out. And it keeps coming out. I'm serious. I open up the science journals every week and I'm just blown away because there's always something new, always something worth learning. They're always answering old questions and generating new questions. Uh, why should I throw any kind of creationist dogma into that beautiful soup that we've got out there in the literature? It's it's just terrible. Anyway, yeah, okay. Big Zebra says, I think of creationists and their ilk as the forces of organized ignorance. Yeah, there's a whole lot of that going on. We got a suggestion here. Maybe there are some ways you could promote your YouTube channel more to shift the balance a bit. Yeah, that's another thing is, do I really want to get into the promotional game? Um, I'm happy just having a place to voice my opinions and my ideas and all this wonderful science information that's out there. And I actually don't feel too desperate to promote myself. Like I said, I'm old and homely, and I'm not going to be the face of science communication. So why should I promote me instead of just promoting some of the ideas? So, if I could do more to promote other science communicators, I would be happy to do that. I link to them on my blog and so forth. But I kind of feel like my role at my age is let's let the kids do it. Let's tell them to get out there and do fun and exciting things, and I'll be inspired by them, and I'll sit by the sidelines and go rah 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 and say do some more. That's kind of my role anymore, I think. and that's okay. Oh man, the light. What is this thing right there? Anyway, yeah, I'm one of the problems with doing this at three in the afternoon is here I am in the northern climes and the sun is going down over that way and it's shining through my blinds and creates some interesting lighting effects, kind of annoying lighting effects. I should probably start doing this later in the evening or something when I can just forget about stupid old sunshine. Okay. Let's see we got any more questions to answer. Invite who to the show? Somebody says I should invite them to the show, but they didn't say who them is. I don't want to invite creationists, but I'd love to talk with other science-minded people. Uh, Shortly before I started this, I got a phone call from Aaron Ra, and I'll be doing a little bit of a show with him tomorrow that will be put on his channel later on. But yeah, don't invite anyone to the show unless they bring snacks. Oh, okay. You meant younger science communicators. Yeah, I'm all for that. But the younger science communicators don't need a fuddy-duddy like me to help them out. They're doing a fine job all on their own. So I'm not going to be so arrogant as to suggest that. Anyway, here we are. Uh, what am I going to talk about next? Well, here's what I thought I'd do today is I promise this is not going to be one of those 3 or 4 hour videos. I do not have the patience or the time for those anymore. Uh, what it is I read this really interesting paper in Nature, and it addressed a topic that I have felt compelled to address in my cell biology course, but I haven't been doing an adequate job, I don't think. And this paper just nicely illustrated a bunch of concepts that I think are important to get across in cell biology. So let me start there. I'll talk a little bit about what I teach in cell biology and why this paper got me enthused. Let's see, I gotta find things. Let's find this. Where'd it go? Is that it? Yeah. Okay. So, what I've just put on the screen, I hope it's working. I Here comes this leg. There we are. Okay. Uh, so what I put on the screen is, first of all, a slide that I use in my classes. So I'm teaching cell biology, and early in the term, one of the things we talk about is these different kinds of cells. And I'm sure you all have heard of these, that we have our bacteria, our prokaryotes. We have the archaea, which are extremely interesting from an evolutionary point of view. And then we have eukaryotes like us. And in the context of this course, what I have to explain is what are the differences between these groups of organisms in the modern day? What do we see when we get on the microscope, when we go look at it, bacteria and archaea and eukaryotes? And so this is just one slide out of a longer series that I won't get into, where I discuss each of the different components, the differences between eukaryotes and these other groups. And the first one I start off with is the one everyone's familiar with, the nucleus, right? That's why they're called eukaryotes, because these are cells with a specific nucleus that contains all the genetic material, the DNA. So that's one important distinction. Uh, Another important one is they have an endomembrane system. That's what they have, these membrane-bound internal organelles. So mitochondria, chloroplasts, endoplasmic reticula, Golgi apparatus, all this stuff that one of the things that's special about eukaryotes is that they've packaged up all of their goodies and their internal goodies into compartments. So that's important. They have this thing going on. I mentioned the flagella because this is a distinct difference between bacteria and eukaryotes. Um, and we have a section in our cell biology class where we talk specifically about flagella and flagellar evolution. Uh, they have ribosomes. All of them have ribosomes, prokaryotes and eukaryotes and archaea. But the ones in eukaryotes are slightly different. They look very different in some ways from prokaryotic ribosomes. So that's an important distinction. Um, then another point we make is eukaryotic genomes are terrible. They're mostly empty, wasted space, just nonsense, garbage scattered all through it, and only a small fraction is coding for proteins. There's a larger percentage that's coding for regulatory stuff. Eukaryotic cells spend a lot of effort on regulating gene expression. So that's a difference. Oh, I skipped one. They have a complex microtubular cytoskeleton. So this is another key feature of eukaryotes is, if you just look at cells in your body, they're all eukaryotic cells, obviously. Um, they have different shapes, different structures, and a lot of those different shapes can be tracked up to the variations in this cytoskeleton. And it changes the way, the shape and the structure of the cell. So they built this really intricate framework on the inside. And then another key point we make, the guy I teach this course with is a cell signaling person. So of course we got to talk about cell signaling, that eukaryotic cells are pretty good communicators. They're all talking with each other. They're sharing information. They're signaling to each other. And they're using that information to make decisions about, secretion, expression, morphology, everything. So that's an important thing in eukaryotes. Okay, so like I say, I'm just setting this up. I've got all these different things that I then introduce and we go through in detail in the course discussing each of these. But I've always worried that the way I present this is a little misleading because it makes it sound like, boom, all of a sudden, eukaryotes evolved all these different features. They're all there in their cells. And it makes it, I'm compressing everything. I'm compressing the timeline to say, okay, we're just worried about modern day cells. So here's all this cool stuff that was going on inside of a eukaryote, and we'll just present it as given. It's there. It's happened. And the question, of course, is how did it happen? This is a complex set of structures. And so then the question is, what order did these pieces arrive at? Which came first, the nucleus or mitochondria? The answer is going to be nucleus. But anyway, um, what about all these other things? When did they arise? Did they all just go poof and there they are? No, of course not. They're all working together and they evolved in parallel over a long period of time. So I would love to see a cell, a eukaryotic cell or a pre-eukaryotic cell from 2.5 billion years ago, because they would be a hoot. They'd have all kinds of differences. And we'd see all these different systems in the process of evolving. But of course, they've all been displaced by modern eukaryotes. You just don't see them now. Okay, so that's what I found interesting about this paper. So I'll just scroll this up. This is the paper. Dated gene duplications elucidate the evolutionary assembly of eukaryotes. So you see the context I have here is, I was saying, here's all these pieces, and then here's this paper that talks about the order and pattern of their arisal arrival in the eukaryotes, going piece by piece through them and trying to put them into a timeline so we can see exactly when things were happening, which I think is pretty darn cool. That sounds really interesting to me. So I won't go too deeply into all the details in this paper, but let's just take a look at what we're doing. So here, if we just look at their first figure. So one of the things that bugs me about every molecular biology paper, and it's especially true here, is acronyms. There are so many acronyms in this paper. Oh man. So it was a little bit of a struggle to get into the language at first. And so I'm looking through this. I actually made a little glossary for myself so I can remember what all these different things were. Okay, but first the method. What are they doing? They are doing this genomic comparisons across a huge number of bacterial and eukaryotic species and archaea species. And what they do is they sequence them and they look for a history of divergence. They're looking for gene duplications. So they look through those genomes and they group them together and they can assemble a diagram like this tree diagram here. Let's see, maybe I can blow it up a little. Yeah, maybe. Oh, but I know I want to scroll and it's being annoying. Um, okay, maybe I won't do that. Back off a little. There we go. Yeah, it's still legible. Anyway, so what they do is they put together these tree diagrams. And the important thing about these tree diagrams is that they can then look at the age of the divergence that occurred in this particular lineage, and they can place it on the timeline. And so we can ask for all these different components. When did they start to diverge? And so, for instance, what you see here is, see, did I lose them? No, I did. I'm fine. What we see here is that we can identify uh, last common ancestors for various features. So that's where all the acronyms come in. So let me explain what these acronyms are. So NFECA, that's the nuclear first eukaryotic common ancestor. So this is when we believe that the first cell made that distinction of a nucleus from a cytoplasm and compartmentalized its DNA. And by this kind of tree analysis, you can figure out when that happened first. So that's when we first get um, a nuclear organism, an organism with a nucleus in it. MFECA is the mitochondrial first eukaryotic common ancestor. So this is where we've absorbed another cell and sequestered that as the powerhouse of the cell. That's when we get mitochondria. So we can ask, when did that happen? And then this LECA is the last eukaryotic common ancestor. So what we're doing is arraying these different species on a timeline to figure out these pieces. When does all this come together? Uh, these are the eukaryotes up here, and down here we have the LACA is the last archaeal common ancestor. So we're looking at the archaea here, and bacterial here. We're looking at prokaryotes here. And of course, prokaryotes are much more diverse and more complicated in many ways than the eukaryotic lineages. And then here, this is a familiar one, last universal common ancestor. And so we're putting all these into this timeline and getting a feel for when various events occurred. And so, for instance, the last universal common ancestor, over 4 billion years ago, closer to 4.5 billion, So really, life just sprouted on this planet shortly after it cooled and water precipitated on the surface, and we had an environment for these organisms to live in. And so this is really old. We got a question here. Is it possible that life appeared more than once and still became the organelles? Uh, no, we've kind of ruled that out because uh, there none of these are completely separate from all the others. So we can find common things between bacteria, archaea, and eukaryotes. So we know they all shared a common ancestor. So that there had to have been one. There was also probably, though, muddle things up. There was a lot of horizontal gene transfer going on all over the place. So it's not a linear relationship between us and our last the Luca. So it was complicated and blurry. And there was this big tank down here. We're talking just a complex spaghetti tangle of all these different species interacting with each other. So, uh, can we say that there was only one origin for everything? Yeah, because all of these organisms have something in common. They've inherited something from a distant ancestor. That doesn't mean that there weren't things that were independently evolved in different lineages, just that it's yeah, if we were doing the genealogy of all life, it would all funnel down into a last common ancestor. Uh, but along the way, there would be a lot of complicated inbreeding and tangling of lineages and novel lineages getting soaked up and absorbed into it. So yeah, we're eukaryotes like us, we have a fairly simple method of reproduction. So that allows us to be fairly discrete and recognize specific ancestors in our past. But that's not true when you get down near the base of the tree of life. Okay, so use this technique to work out all these relationships and figure out, for instance, here we've got um, these different lineages here that are making nuclei, and that arose right here. I don't know if do you see my arrow when I'm wiggling it around up there? I'm just imagining that. Anyway, so question, mitochondria and chloroplasts are at least different, right? They're still related. Mitochondria and chloroplasts are of prokaryotic origin, but they also rose from this last universal common ancestor. So um, they're different, but they're still part of this family tree. It's like, if you could go back and you could find your distant third cousin and absorb them into your body, but they're still your relation. You still can trace to a common ancestry. You still share the same great 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 grandfather, for instance. Okay, so this is their technique, is they're just going through all these and building up a catalog of all these different organisms and piecing them together and trying to figure out how these different components arose. So, like I showed you up here, I said we got these multiple components that have to come together to make a eukaryote. They're going to ask for each of these, when did they arise? How did they arise? What was going on there? So for jumping ahead, for instance, one of the things they look at is the evolution of the cytoskeleton, microtubules and actin. And they can look at these duplications that occurred in the genes that make these structures, and they can ask, when did all this arise? That's what these funny little diagrams, these little curves are for, is these are estimates of when the the particular detail arose, and there's a key down here. Distribution shows the pre-LECA last eukaryotic common ancestor, duplication of the different nodes. And you can see 95% confidence interval. That's what this band here is. So we can leave an idea roughly with our confidence intervals. We can get an idea of when these things arose. And one key and obvious point that as I was complaining about earlier, I don't make clear in my cell biology lectures is that these didn't arise all at the same time. They put together this progression of changes over time that gradually added new capabilities to the cytoskeleton. So we got that going on. Here, for instance, actin, one of the key things they're looking at is um, making forked branching patterns in the actin cytoskeleton that required these particular molecules here to arise. When they arise, well, we look at them and we can figure that out from these comparative studies. So we're adding this capability instead of just linear girders everywhere. Now we can have branch girders, and that allows new variations in the cytoskeleton. They're also looking at things like or polymerase, RNA polymerase in this case, and we can see that these different polymerases arose at different times, and they're specialized for different functions. So, for example, POL2, I think it's POL2, is the one that's primarily involved with things like um, splicing. So it's making SNRPs, single nucleotide polymorphisms, and proteins that work together to splice together pieces of RNA to assemble a complete end product. And so that was arising back in here. Okay. And you can see from the timeline, roughly 2.5 billion years ago, this is happening. And then there's these specific structures, the SNRPs that are involved with the splicing itself. And we can see when they were rising. So there's all this stuff going on well before we have what we would classically call a eukaryote. And they're rising gradually over time. Yeah, that's a good word. So Hemi Scott mentions foreshadowing. That what that is, we have molecules arising that foreshadow greater capacities for eukaryotic behavior and also for things like cellularity. So this is the way evolution works. You assemble these pieces that in their initial incarnation are just refinements on an existing process. And then they are later going to be opportunistically used for even more complicated, sophisticated, interesting things like multicellularity. We'll consider multicellularity in this paper, but that's eventually going to come along and it's going to take advantage of all these features that are sitting there. Uh, okay, Hemi is also interested in metazone cell adhesion. Oh, that I don't think the paper gets into that very much, does it? The paper has a lot of details, so I may have missed something. But like here, they're also looking at things like the endomembrane system. Oh wait, I got a question here. Yeah, and I think two and three do some split. No, they don't do the splicing. They are specializing for producing the gene products that then do the splicing. So we have one set of phrases that are there you know, to make copies of the conventional genes we all think of when we think of gene products. They're making structural genes and enzymes and all this kind of cool stuff. And then we got these other specialized polymerases that are dedicated to just producing these internal processing components for things like splicing. Yeah. Okay, where was I? Which one does the better job of splicing? Yeah, neither of the none of the polymerases do the splicing. It's all the other stuff that's going to do the splicing. These small nuclear proteins that are going to get in there and splice things together. Okay. Oh, and this is the slide. We're asked, how do vesicular and protein intracellular traffic evolve? I mean, complex interaction and protein processing from endoplasmic to Golgi apparatus to endosomes and lysozyme and so on. Yeah, that's what this slide is all about. They're looking at these different components that had to have evolved to produce the endomembrane system. So we're seeing this gradual specialization of different elements of this endomembrane pattern to do specific functions. One of the things that this we think is responsible for, as it's mentioned here in this line right here, uh, the autophagosome. So this is basically the gut of the cell. It's making a digestive system. So we're going to have little temporary vesicles that form that contain engulfed material. And then we're also going to build things like lysosomes. Lysosomes are packed full of enzymes and various reactive products that we can then fuse with a vesicle, and that will then break down the contents. So it really is kind of a digestive system that's right here. So we're seeing all that evolving in here. And again, you can see this is happening early. Remember, last eukaryotic common ancestor is over here, and they peg it at somewhere around 1.75 billion years ago, somewhere in there. And all this other stuff is happening in the diverse pre-eukaryotic organisms that are thriving in the ocean way back here, all the way back to 3 billion years ago. And they're just assembling the pieces to more efficiently package and transport and digest various components. So that's all happening gradually and piece by piece, assembling together to make this endomembrane system. See, would you say a stable environment or a changing environment is originally inducive to evolution to a new species? Also, I'm curious about hybridization as a major part of speciation. Yeah, both. We need both. Uh, changing environments provide new opportunities and give variants an opportunity to expand. But you also need stable environments to some degree so that populations can expand and build on prior structures. And then this is from an era when lateral gene transfer is commonplace. You know, it's commonplace now among prokaryotes. So this would have been a time when all these differences could evolve in independent lineages, and then they could be transferred and shared to each other. And so you get this gamish of all these different structures emerging over this period from, like it says, about 3 billion to 2 billion years ago. Okay, so that was another component is building all this stuff. And then here, this is all related to the pre-adaptations to allow for the emergence of mitochondria. So we first have a system where we've got a cell that builds a nucleus, and at the same time, it's also adapting its endomembrane system to do these complex interaction, things like digestion and packaging and transport and all this important stuff going on inside these cells. And then only later we absorb on another prokaryote. We take that into the cell, and now we're going to start making adaptations that allow it to uh, to survive and be opportunistically used in the cell. So a whole bunch of stuff going on in here. And these, again, we're still looking at the same timeline, 3 billion years ago to 1.75 billion years ago, is all these changes are going on at different times, different places, and they're all making this organism copacetic for this kind of incorporation of prokaryotes. Okay. Let's see. The cells, another question. The cells do use splicing for cell division, right? Uh, no, no. Splicing is something you do in the production of RNAs. So there are some of the splicing enzymes are adapted for repair of DNA, but in general, no, these are not relevant for cell division. It's only when you completed division and you started making proteins. Uh, remember that sloppy eukaryotes are our genes are broken up into exons and introns that all have to be stuck back together appropriately to build the complete final protein. So yeah, um, that's a different thing. Okay. Phagocytosis is going to make them susceptible to invasion by bacteria. Yeah, and Legionnaire's disease carries this susceptibility into human macrophages. Um, yeah, so this is this is one of those things why this is one reason why it was complicated. You just don't eagerly allow a foreign organism to come in and settle down and invade your tissues and thrive there. That's the pathway to parasitism. Uh, so that's one of the reasons we got all this stuff going on in the pre-adaptation of the organism is you got to figure that a lot of there were a lot of bad starts here where yeah, we let in some organisms. Uh, then we had some problems where there were organisms that would take advantage of that to eat you from the inside out. So we got all these different features that have to evolve gradually so that you minimize that risk. Okay, RNA for cell function. Yes, that's where splicing is important. And you get them out of the DNA in RNA form. And because of the way our system works, we get the RNA from the DNA in little splice chunks. So they're all kind of scattered together with gaps in between. And then we've got to have these splicing enzymes that snip out the garbage and stick them back together to make the whole sequence of the final protein. So that's kind of what we're doing here. Oh, let's jump ahead because I really like this particular slide here. Um, this is a useful image. I might not use a lot of the other stuff in my class because this is a sophomore level class and you can't charge into all this stuff until you've spent a few weeks explaining all the bits and pieces. But I thought this was pretty good, especially this bottom figure. Um, what it's basically showing is here are all these different components being a eukaryote, all these little pieces that we have to assemble to make a full eukaryotic organism. And we can see this like here, we start with membrane fusion budding proteins. So we need the cytoskeleton for that. So we're going to start doing that. Uh, then we're going to build with the cytoskeleton, we're going to build tubulin, we're going to build actin, branched actin, etc. So we're going to be able to assume different shapes. Uh, in eukaryotes, we have these microtubule organizing centers. So we build these little structures from which the tubulins emanate. So our skeleton, our cell skeleton has little joints and structures to hold it all together, things like the MTOC. And then that's all getting incorporated into gene trafficking families that are then going to be important in the function of the endoplasmic reticulum and the Golgi, that are going to enable us to make lysosomes and endosomes so we can start taking things that we eat. And when I say we, I'm talking about primitive cells, just single cells that are doing this. We're bringing all this cool stuff to have for dinner, and we got all these tools now to digest and break them down. Um, we're also using this endomembrane system to build the nucleus. The nucleus is just an encapsulation of a bunch of inserted membranes that wrap around the DNA, making a localized vesicle. And we got phagocytosis going on. Uh, it also mentions horizontal gene transfer here. And we got membrane transitions. We're going to build all these specialized karyotic uh, membrane lipids that are going to do interesting things there. And then we're ready. We can bring in mitochondria, the endosymbiosis. We're making mitochondria, and that's then going to eventually lead to meiosis, which means sex. Sex comes after all of this. Okay, we got some comments here. Many metabolic pathways, mitochondrial like yours. Oh, yeah. So a lot of mitochondrial function is related to cell death. That becomes particularly important in multicellular organisms that are that you've got all these different cells cooperating, and you got to have a mechanism for policing all of this. And one of the things evolution has done is it taken advantage of these mitochondria, these separate little organelles. So they're also a source of signals that can be used for triggering cell death. Okay. And yes, Adrian, all of this is inside of us, inside of all of our cells, each of our cells. It's inside of things like paramecia and amoebas too. And of course, it's healthy. That may be a little too glib that it's healthy. Because when things go wrong in all of these pathways, that can lead to things like cancer. So it's this elaborate house of cards that we've constructed to get this sophisticated functioning out of our cells. And if things go bad in some piece of it, the whole thing can collapse. And then you got a dead cell. That's one of the reasons why, as we just mentioned, um, mitochondria were enrolled in the job of also acting as little assassins. When things go wrong, we have to kill that cell before it makes everything worse for everything else. The thing is, of course, if we got a multicellular organism, we got all kinds of redundancy. We got all these duplicates that we can take and use those. If one of the cells is lying down in the job, you can go over there and kill it, and some other cells will take over. In some ways, it's kind of a ruthless system. But at the same time, it's a system that's fairly tolerant of error. We're all full of individual somatic mutations that can do terrible things, but usually don't. Occasionally they'll do horrible things, but usually we get by just fine. Okay. So that's the story. That's like I said, in my cell biology course, I could see using this because what that does is it takes this rather blah text slice that I use right now, and I could then use that diagram to illustrate all these different pieces and how they're not just arbitrary, they're interrelated and they're all necessary for the function of the cell. So yeah, it all fits. Means I get to add some new stuff to the biology this coming year. At least the cells can't feel pain, according to Jeremy Bentham. Okay. Is that what is pain? I mean, I could have you ever watched a cell die? It's pretty grisly stuff where they just you can see everything kind of coagulating and breaking up internally, and then boom, um, it all goes squirting out and flying around. And I have literally seen cells trembling on the dish that then go kerboom and explode. But of course, they don't have brains. They don't have senses. They don't necessarily feel anything. Yeah, genetic drift happens in bacteria too. It's populations. So we're looking at population functions there, not individual cells necessarily. I wonder if they do have emotion. Oh, let's get philosophical here. Do cells have emotions? And I I don't again, they don't have a nervous system, they don't have a brain, they don't have anything to think with, but they do have an impulse to survive. So you could imagine a cell feeling the rudimentary perception of disappointment that, oh, I'm blowing up. But I'm not too worried about their emotions. Okay, as I said at the beginning, I don't want to be long-winded and go on for hours. So I'm gonna draw this close right here. Any last questions? I don't know when I'll do this again. I always say I'm gonna try and get on here more regularly, but then I never do because I always get distracted. I got more important things to do. I can say I'm going to be recording with Aaron Raw tomorrow, so sometime the next week you can see me again if you have to. Um, yeah. Okay. So genetic drift is not genetic information traveling between organisms. That is correct. It is not. But that would be horizontal gene transfer. Another question. Oh, they're coming fast now. Does gene regulatory network control gastrulation, cleavage, organogenesis, or even the brain network? Well, yes, of course it does. Yeah. Um, there's a huge amount of developmental biology literature precisely that. Think of things like Eric Davidson's work on differentiation of cells and echinoderms, that there are these gene regulatory networks that take in information from the environment, process it, and have the cell make decisions about cell fate and cell patterns of movement, cell patterns of activity. So yeah, it's all happening all the time. Okay, or did emotions evolve from cells having different metabolic states for different conditions? That's a mechanistic thing. Um, emotions uh, emotions arise from the complex patterns of interactions between numerous cells in your brain. And yes, those have different metabolic states. They are producing different kinds of transmitters, etc. So yeah, that's sort of true. But again, we wouldn't talk about emotions in a eukaryotic cell from 2 billion years ago. It just wouldn't apply. Because at that time, they haven't built these complex networks where you can have multiple kinds of physiological interactions between cells that you need to produce things like emotions. Is the Aaron Raw thing? No, I think Aaron and I are going to record something tomorrow, and then Aaron will assemble it into a video that gets posted on YouTube. This is also something I find annoying about YouTube's method of doing things. This particular video will not get much attention. It's going to be promoted by the YouTube algorithm because it's a live stream. Uh, it would be better if I recorded something, assembled it digitally, and then uploaded it as a package all at once. Then YouTube would like it better. So it's a packaging thing that we have to worry about. Um, let's see. We're getting into some speculation way. I could imagine different kinds of whales having a brainiac evolutionary arms race going on. Well, yeah, whales are pretty smart animals. We don't know why. Um, Another thing that might just be going on is just as a passive consequence of expanding the size of their brain as part of growing larger to feed in their environment, they acquire new capabilities that can be used in intelligent ways. It doesn't have to be, intelligence doesn't have to be a directed goal of the organism. It can arise just poof, spontaneously from things like increasing complexity of the nervous system, which is actually a terrible thought to think that, yeah, some little organism that's all its only purpose is reproduce and dine on algae or whatever. And as evolution occurs, suddenly it's sitting there with the ability to do poetry. Um, let's see what else we got here. Yeah, I agree, Emmy. Emotions are more complicated states in brain having organism in mammal and birds. Spiders probably have emotions too, though, I would add. Okay, you're studying Gilbert's developmental biology book. That's a good book. I like Gilbert. Um, I'm not teaching developmental biology. I will be teaching um, Eco Devo next year, and I will use Gilbert's Eco Devo textbook. It's a really good book. Okay. Goldie's 1464, Mrs. Peasy's atheist rants, but your science posts are great. Oh, thank you. I'm glad to hear that. You know, when I'm sitting here home alone reading some of this stuff, I do rant at the screen when I see these morons getting up. I mean, there's this whole mafia of Christian apologists that that just make me want to tear my hair out because their arguments are so bad. They're irrational. There's a complete dearth of evidence behind what they say. And yeah, I sometimes feel like I should just get on here and yell, but I feel like I'd be incoherent and angry. Nothing wrong with that, though, of course. Yes, I've used the older Gilbert text in my class in the past. Uh, I think I've got his updated version somewhere around here. Oh yeah, okay. Adrian mentions, I've once heard the thesis that human intelligence came from a sudden brain expansion. Yeah, that's I think that's a viable hypothesis. That something just switched on increased mitoses in the developing brain tissue, and we get all this stuff. Of course, brains are also really expensive organisms or organs. They consume a lot of energy. So there that would have to evolve some kind of adaptive value fairly quickly. Otherwise, yeah, you know, Why keep this complicated structure that requires a lot of metabolic energy to, you'd have an advantage if you developed a tiny brain, which explains the evolution of Christian apologists. Yeah. Okay. I think so John St. Francis says, I think emotion on a more primitive level is earlier than brains. And I agree um, that a lot of the drives that keep an organism active and doing things and going out and mating and risking their lives for food, that's emotion. Thing, rationality and thinking and planning on top of that just just fine tunes the expression of those emotions. So yeah. Um, oh yeah, the apologists owe all their social status to the BS they teach. That's one of the problems. I agree. I've watched some of these apologists and it's appalling how bad they are. Uh, they just don't have a grasp on basic logic, but you can see that they're just doing their best to defend a prior belief that they have acquired, and they don't question the dogma. So they got to keep building up excuses for it, and it's painful to watch. Okay, another good you. These questions are never going to end at this rate. Do you think Evo Devo should be represented as an extended version of evolution? Um, I don't even know what that means. What do you mean an extended version of evolution? It's a part of evolution. Develop Development is one of those things that organisms go through. We know, for instance, that there are different evolutionary pressures on a larva versus an adult. They're both evolved to perform particular functions. So yeah, it's kind of an important thing. Do I think consciousness is unique to humans? No, not at all. I have a cat somewhere around here, and that cat is conscious, and she is selfish and whiny and really obnoxious. Um, I think there's definitely a discrete, separate consciousness driving that behavior, even when it irritates me. Okay. Is that it? Are we done? Should we wrap this up? I think it was fun. I had a good time. Maybe we'll have to try it again another day and see what comes of it. Maybe Aaron Ra will inspire me to some things. Yeah. I would accept a person uh, three times as much if they're mistakes, but not all people are like that. Yeah, the kind of stupidity we get from people like Trump, who would never admit error, even though he makes so many of them. That's how we can never get better. Uh, Hemi Scott agrees that I'd say many animals have consciousness. The trickier thing, self-awareness. Okay. Yeah, and I have thought about that too. I mean, there's things like the mirror test to see if they can be aware of their status. But I think just, self-awareness is such a complicated, messy topic that who knows? Maybe there could be different kinds of self-awareness. Okay. Oh no. Last question. Do we have free will? Oh, you're trying to annoy me now. I despise the question of free will. I don't believe in free will. I think it is an invalid concept that we waste our time pursuing, that it's meaningless. What does it mean to have free will? No one can define it. Uh, no one can explain the context to it. So, yeah, I am extremely dismissive of free will questions. And yeah, I know some people make a living practically arguing about free will, but I just can't see it. Okay, this will never end. So I'm going to close it here. I've been on here for 70 minutes. That's over the hour limit I usually impose on myself. So I'm going to end it now. We'll talk to you all later sometime, I hope. I'll, if I can get over this crippling self-doubt and get my face on the screen again, I'll try to do it. So talk to you all later.